I'm a little overwhelmed all of a sudden and kind of a little nervous, even though I didn't think I would be. My son asked me yesterday if I was nervous, and I said, no, not at all, but now I'm shaking. So I have half an hour, right? Because this says 9 minutes, and I know that I don't have 9 minutes. Okay, thank you. All right, so I do, you know, when you get in a word like this, you do get a little introspective and try to think, and you think a little bit about your life and where you are and what you're doing. And I was just telling someone in the back of the room, I think I'm going through a midlife crisis as well, so this couldn't have come at a better time. Kids are empty nesters, and I mean, my kids are gone, so now I'm like, I can't work all the time, can I? And you can't work all the time, it's not good for you, I'm realizing that. Sorry, now I'm rambling. So I did want to just start off with some disclosures and thanks to people who really helped me through the years as a way to start the lecture rather than the end, because I'll probably go too long like I normally do. I do need to thank the NIDCD. They have funded our work since I graduated with my PhD in 2001. The first grant listed has been funded since 2000 and is specifically on tissue engineering of the larynx, which was awarded to Steve Gray, and then I carried that forth, and we've been going now on for over 25 years on that grant. The NICDCD has been nothing but wonderful. Certainly there's always the bad grant review, but other than that, it's been a great ride. Lana Sheckham retired last year, and she had been my program officer for 20 years and was nothing but fully supportive of the work that we do. My mentors, I mean, there's just too many, and I can't really thank them all. I do want to highlight 3 people. However, Bob Zaveloff. My first Voice Foundation was in 1999. I was a student. I did a poster. I was a baby, and you have been nothing but supportive of me through the years, pulling me aside, giving me advice, comments, occasionally even giving me a call, and I've always been very appreciative of that support. Thank you. Steve Gray, who we lost in 2002, really lit a fire and was so enthusiastic about this type of research. We lost a pioneer way too soon, and I am honored to carry on his grant. And of all the grants I've had, it's always been the most important one to me, so thank you. I just lost my glasses. Oh, they're on my head. But far not least, I am blessed, and who's winning the Soonberg Tietze Award tomorrow, Lifetime Achievement. I met Diane in 1997. She took me on as her graduate student after me having to email her about 3 or 4 times. It's a big joke, Diane, where are you? And since that time, she's been my academic mother. She's been a mother to me. She's been a grandmother to my children. She's my next door neighbor, but we live across the street from each other. Really, a neighbor, a colleague, and a friend, and I really just cannot thank her enough on a daily basis. We talk a lot, and even through the years, now she doesn't want to talk about voice. You know, hey Diane, will you give me some advice? She said, no, she's busy, she's got stuff going on, but really, she has been a fundamental foundation in my life, and I'm so grateful for our relationship. Thank you. I don't know where you are because I can't see, but, and wave, where are you? There's Diane, thanks Diane. I've had a tremendous number of mentees and trainees in my lab, and I put this quote up here. Working in my lab is not for the faint of heart. I have a tendency to push people, and I push them, and I have very high expectations because I believe most people can meet them, and if they're having trouble, we try to remove the barriers to make them successful. And I judge my impact by how successful my trainees and my scientists are, and I'm forever grateful for the work they do. I could not grow a cell now if you paid me. Wouldn't even know how, I would kill it. They do everything in the lab, and I am tremendously grateful. I am tremendously grateful for this award and the committee that chose me. I'm the 23rd person to get this award. I am the first female to get this award. Many more to come. So I throw that gauntlet out there. The world of vocal fold biology is bright, and there's a lot of work to be done. So I'm grateful, and I look forward to all the other women out there who succeed in following my path. So thank you. All right, so let's go to some innovative fun science, okay? And I'm going to try to present some new stuff that we have that's not published yet. I try not to present stuff that's not published because people who know me well say if it's not published, it's just gossip because it hasn't gone through peer review yet. But I figured out since it's the Quintana Award, I would show the most fun, innovative stuff that we're doing that I think has potential to change or guide many people in the work they're doing. So what is tissue engineering? And it's an area that I've been studying with since I started my PhD. It is an interdisciplinary field. Started in 1988 at MIT with Langer and Vacanti, who termed the phrase. And it is applying the principles of engineering and life sciences towards a development of biological constructs, tissue, organs, etc., and to actually also regain function of those entities. So as a construct for the lecture, I'm going to talk about tissue engineering, what we're doing as an application for tissue engineering, specifically of the larynx, and more specifically for the vocal folds. So I've been doing this for a long time now, it seems like, and mind you, it goes really quickly, but there's a lot of challenges in tissue engineering of the larynx, and people have had some people say, well, how come you don't have anything yet, Susan? And I just, yeah, I wish it was that easy. But so the larynx, as we all know, is super complex, right? I don't need to tell you guys. There's muscles, there's tendons, there's cartilage, there's all kinds of cells. I mean, there's just it's complex. It's comprised of many different cell types and various entities that also have to function. It functions as cartilage does. It functions as really dynamic vocal folds that that vibrate faster than anything else in the body, and trying to regenerate that kind of tissue is difficult. We have to maintain function. We can actually develop almost anything for the larynx. I'm going to get different glasses, I'm sorry. We can tissue engineer anything. The issue is, can it actually move and function as we want it to? And we can't do that because the tissue is incredibly delicate, and if we put something in the larynx that's Teflon or like cement, it's not going to function well. It may actually structurally be sound, but it's not going to actually function. Sorry, guys. All right, much better. So the larynx also has a very strong vascular system, and it's very hard to regenerate tissue that has that vascular system, or to make something that the vascular system can go into and actually provide the nutrition that it needs. And last, it has to be something that's not going to be rejected. So we know that if we put anything in the body, there's a chance that it's going to be rejected, and that's a pretty big issue. If anyone has ever been abreast of what went on in the tracheal literature, we know that they deconstructed some tracheas, slapped some cells on it, and then put them in a bunch of patients. And now, years later, the folks involved have even gone to jail for what they did because they created such harm on patients for their lack of insight and whatever. I don't know what they were charged with, but certainly they did it in a way that seems like it was super easy, but indeed was not. And bodies rejected what they made. So what are the practical limitations specific to the larynx that make it even harder for us? Is that we don't have available tissue. If someone is doing heart research or lung research, it's pretty easy to get hearts and lungs from humans. It's really hard, or even pieces of tissue biopsies. It's really hard to get vocal fold tissue or larynx tissue. We can certainly get larynxes from cadavers and decellularize them. We can certainly get small biopsies of abnormal tissue. I can't take a biopsy of anyone's normal larynx. I can't imagine anyone here in the room is going to let me do that. So we have to really try to understand what we're doing without having a free source of those entities. There's not cell lines available. If you're doing lung research, there's a million companies that sell cell lines, and you can go online and click the button and get them. We can't do that for vocal folds. We have to make them ourselves and share them. And when you make them, you have to be really rigorous to show that what you're making is really what you say you're making. We don't have an animal model that's ideal. We're the only entity that communicates the way we do and uses our larynx the way that we do. We certainly try to use a lot of different animal models, and generally we use them based upon the assay we have, but there's not an ideal one, and that's problematic. And grant reviewers, if you're a grant reviewer, there's no such thing as a perfect animal model. Please don't hammer somebody on that. There's no such thing. And, you know, if people can justify it the way they do, I think that's great. And that's been a huge detriment to the field. It's a small field. There's probably anywhere between 5 to 10 of us that do this, if that many. And you know, a field's only going to get good and be productive quickly if there's more people doing it. There's just not enough of us. There's some collaboration, but it's hard, right? There's only so much to do, and there's only so many things. There's so many questions, right? So we can only do much. And then we have a really poor understanding of the basic biology of the larynx and the vocal folds. And certainly, we know more than we did 25 years ago. We're still a long way off and have a long way to go. So those are the entities. So the complexity of the field is daunting. Things that have been done to date. We know that vocal fold cells respond to vibration. There's been lots of bioreactors made and vibrating cells in different ways. I've done that. There's lots of biomaterials and gels that are out there. Everyone's trying to make a gel, gels, and more gels. I did a lot of the same. There's synthetic, natural, hybrid. They put different types of cell in them, inject them into the vocal fold, and see how they do. And then there's been the idea of just taking the larynx as a whole. I'm taking all the cells out and then putting that back into the body with cells of choice. Pick a cell, any cell you want. But all of this is done quite randomly because of the complexity of the field. So what I wanted to do today is talk a little bit about the most recent stuff we've done. And I've been kind of known to be a little bit attention deficit, for lack of a better word, where I like to follow the technology. I get bored really quickly. So I follow the technology and what's being done in the field as a whole and bring it to the larynx. And I've tried to build a foundation. So knowing that I, for example, may not ever find the right thing or tissue engineer the larynx, but can I provide a foundation that other people can use in the future? For example, we have about 11 cell lines of vocal fold fibroblasts, the most in the world in my lab, and I share them openly and freely to anyone. You just have to pay for shipping. So things of that nature, we just, you know, we build a foundation. So there's a technology that's come out more recently. It's probably about 5 years ago called single cell RNA sequencing. And up to this time, we've been able to take tissue, mash it up, and say what is being expressed by the genes in this tissue. This technology is more advanced such that we can actually get every single cell in the larynx or any tissue of choice, and we can see what that cell is expressing and what that cell is. It allows us to look at very complex cell populations at once and their transient states, meaning if you look at development, you can see what cell actually becomes a different cell, or what's the precursor to a different cell when you look at development. We can look at how cells are differentially expressed and the trajectories of growth and how they change across time. So people are making cell atlases, and if we think about this in terms of the larynx, if we can make an atlas that says, this is every cell in the larynx and this is where it is, people can use that data to reference and use it for biology, they can use it for disease modeling, they can use it for regenerative medicine, and they can use it specifically for tissue engineering approaches in the larynx. We can also, if you're doing it in different animals, you can cross-reference that to human if it's been done. So to date, no one has done this in the larynx of any human or any animal sort. So we started this looking at, okay, what are the gaps in knowledge that we're going to try to fill, or the holes we're going to fill. And that is, we wanted to characterize all the cell populations in the larynx that reside and migrate in the larynx and the vocal folds during embryonic development, so before birth. And then we want to look at it postnatally through the aging cycle. So we wanted to do from inception to death. And what are those cells that are in the larynx and how do they change dynamically across time? And the idea that people can use that understanding and signatures and knowledge to guide tissue engineering. Because if you want to move a stem cell from one thing to another, you have to know what signals to give it. And we don't know that in the larynx. We know a little bit for the epithelium, but that's it. So, and we also want to look at those relationships between cell types and across time. So single cell sequencing is a process that's, number one, very expensive, and it's not for, again, the faint of heart. So you get a sample. So in this case, we had a larynx, and I'll talk about the study we've done. And You process the tissue where you dissociate it into single cells, you send it to a really awesome biotech a group at your university. Most universities do this now at very different levels, of course, and they process the tissue for you, and they go through quality control, and they sequence it, and then you have this massive data set. Massive. And you then need a really talented, amazing biostatistician who specializes in genetic gene expression data, and we're really lucky in Madison to have multiple. But we're able to get a, let's see, go back one. Oh, let me see. We're able to get an understanding of how the cells are related to one another. We can cluster them by the types of cells and actually start naming the cells in the larynx. We can see what they're expressing, and then we can look at those clusters again and say, are they different within each type? So that's where it gets really fun. So this is a study of one of my amazing grad students who just finished at Lunga. He did this as his dissertation. He just presented it like within last month, where, again, we wanted to look at inception to death in a mouse model. What are the cells present in the larynx? So we looked at important timeline in embryonic development that we know are important for the development of the larynx. We looked at 5 time points, and then we looked at birth, and then we picked times up to 1.5 years. Each of these represent a time where we took the larynx or the vocal folds from the animal. This includes taking, as you can imagine, taking an embryonic E11.5 embryo from a mouse mother and trying to get the larynx out with that involved. We did that. For the young time points, we had to pool the larynxes because they weren't big enough to get enough cells. For 8, and as we were older, we could bring it down to 6 pairs to get enough biological replicates. So the tissue was collected at given time points. It was dissociated. We did the sequencing and library preparation, and we did the data analysis. So what I'm going to highlight is 4 things that we did, and some of them, because of time, I may skip. I'll go with ones that I find the most interesting, or maybe because that's the area that I'm interested in. But if there's anybody who wants to talk about the other stuff, happy to talk to you about it. But we talk about cell types, how the cell and gene dynamics across the lifespan change. We're going to do some trajectory analysis where I can show you what one cell becomes another, and then some secondary clustering, which I think is super interesting. So this is called the UMAP, and this is how they take the data and they say each one of these dots is a cell, and this is all the dots, all the cells in the larynx from birth. So from 11.5 to 1 point year, and this represents all of the cells. And they're color-coded by what cell type they are. And we actually found 23 different types of cells. And we clustered them, in this case, by the epithelium, mesenchyme, immune cells, endothelium, neuronal cells. It's a little bit, the term used during Ted's dissertation was gerrymandering. We live in Wisconsin, we gerrymander us. That was a good one. It's not important how they're related to one another. It's just more important that you understand that they're groups. So all the mesenchymal cells have similar gene expression, and every one of those numbers represents a cell type. And we can we'll go over the different types we found. So these are all the cells, every single one, which is really fun. Okay. So we're able to look at major cell proportion dynamics across the lifespan. So how does the proportion of the cells change over time? And I have a nice red line there because the difference is that from E11.5 to P0, we took the whole larynx. From 4 weeks to 1.5, it's just the vocal folds. And in retrospect, I probably would change that, but it's done. I think a single cell cost us $80,000 just to run thank you NITCD, just to run at the Biotech Center. So we're not going to go back at this point. But you can see over time how there's different dynamics in terms of the cell populations, in terms of broadly epithelium, mesenchyme, from the other types of cells. So there's definitely a change in representations and over time. So here are the 23 cell types we characterized, and we divided them again by their main groups. But in the epithelium, we found 7 different types. Mesenchyme, and this is over time again, 5 different types 4 different types of immune cells and more neuroendothelial and endothelial cells. My lab is always more interested in epithelium and mesenchyme, so that's what I'll talk about, but we have great data on the other ones too, which all of this data will go into a bank online that you have to do, and then anyone, anywhere in the world can access it and pull data and use it for their research, which is one of the beauties of this and being funded by the NICD. So we're able to, this is a dot plot that looks at the 23 types of cells, and on the top are the genes that differentiate these cells from each other, and these are the top 5 genes for each cell type. And you can see there's a nice, so the basement membrane proliferative cell, progenitor cell, is different a little bit from the, I'm shaking, from the proliferating basal epithelial cell, from the non-proliferative. So the epithelial is actually, the epithelial layer of the vocal fold doesn't get enough attention, actually. It's quite complicated. So looking at the epithelial dynamics across the lifespan, we can see the different types of cells, and how they change over time. And for example, we have a lot going on here with the basement membrane producing cells. So these are the cells that actually make the basement membrane. So you can see how many embryonic time points is quite prevalent, but as you get older, or I mean, once the animal is born and you get older, it's less active. And you get more activity, say, of these other types of cells down here, like the secretory cells get much more active when you're aging. So we can see that, and this is just an example of the proliferating basal cell too. These are important because these actually make that very bottom layer of the epithelium that actually makes all the epithelial cells, and so those are important. And we can see from this, this is a heat map, just to orient you. These are the 50 top genes that are expressed by these 2 cell types across time. And what's blue means it's not, the gene isn't being very well expressed at that time point, versus red, which means it's being really highly expressed. So we can say that if we look at a couple of these time points, this is a great example of when here, at very early stages, these genes, the keratins that make the epithelial structure really stable, aren't really involved. Instead, you have these genes that are involved in cell division and proliferation. So embryonically, you're making those cells, right? And then later on, those turn off, and now the body is working on how to make that tissue stable. So, and we can do this, for every cell type. I'm looking at the clock, so I want to make sure I don't go over. And we do this for every type. So proliferating, again, you get a lot more later on. Your body is, your vocal folds are happy, you want it to be stable, you want, again, structure and function, and the suprabasal cells are the very top layer of the epithelium, and those are important because you want to have antibacterial components, you want to have sensory components, you want secretory function, and you're able to get that. And you can see, for example, the suprabasal cells are not very active. They're right here, and look at all the nice blue. Very embryonically, I mean, they're just not, because they're not actually, to be honest, the suprabasal cells aren't there embryonically. They don't actually become, I mean, they're there, but they're much more prevalent postnatal, and they're involved in structure, function, antibacterial presence, and chemostasis, so that's really important. Okay, so quickly, mesenchyme, similar data we can pull. We can look at fibroblasts, which are my initial love. We see fibroblasts throughout the entire time period, and it's the predominant cell. So I'm going to skip ahead because, anyone, if you want to see the details of all of these after we can. Trajectory analysis. Can we actually see how the cells are related to one another? And this is called the pseudo time trajectory analysis. So when you look at the colors are important. That 0 is the undifferentiated cell, that is the initial or the precursor. And as you get to more yellow, that's the more differentiated or mature cell, and we can look at that. So we can look at that with the epithelial cells, and I'm just going to show you panel A. This is a proliferating basal cell, which is red, which is right here. And then you have the non-proliferating basal cell, again, these are in embryonic, moving to a suprabasal epithelial cell, right? That's here. And if you look at the actual trajectory, you see that the undifferentiated cell, which is the proliferating basal epithelial cell, it actually becomes the non-proliferating basal cell, and ultimately becomes that suprabasal epithelial cell. So this is kind of the youngest of the cells, and they eventually develop into these cells over time. And you can also see this with the genes that are being expressed, because as you get a more mature cell, you're going to have the keratins, which actually make the epithelium, versus a P63 gene, which is at the very bottom basement of the epithelium, which actually makes the rest. So this is really great, because now if people want to make epithelial cells, they can use this information to drive stem cells into those, because we can't buy epithelial cells to regenerate tissue. With this data, we can use that to make those cells. And we can do the same thing with the mesenchyme. And again, I'll just do the fibroblast one. So we have the early proliferating mesenchymal progenitor, which goes to a stromal cell, which then becomes a fibroblast. And if you look down here, the purple is the oldest cell and the most undifferentiated, and it moves, becomes a stromal cell, and then becomes that fibroblast of interest. And again, the upregulated genes at the end are the collagens, the fibroblasts are working, versus the downregulated genes that are more interested in just proliferating the cells of choice. Because when you're in utero, you're just making those cells to actually make the vocal fold. Most of the vocal fold doesn't really become developed till after birth, which I don't think is new information for most of us. Okay, so quickly, I just wanted to say that then what happens is that the next thing you can do is you can actually then take the cells of choice, and this is which I think is probably the most fun of a lot of this, is that, so we have a fibroblast cluster, and our statisticians redo clustering, and they say, are any of these fibroblasts different from each other? And they use an algorithm, and we can set it at different resolutions, and you kind of find a sweet spot between too many and too little. And in this data set, we found that there's actually 6 different types of fibroblasts in the vocal fold. You know, those of us who do fibroblast biology think there's one, and maybe that's why we haven't been super successful, because if we think there's only one, and that's what we're growing, and we're putting it in the vocal fold, that might not be the one that we want to grow, right? Like, this is kind of mind-blowing to me a bit. So there's actually, in this study, we found in the mouse, 6 different types, of fibroblasts, and they're here, and you can see the clustering, and you can see over time, this is the proportion to time points again, and we didn't call them anything special other than 1 to 6 right now, but you can see, like, here's one that kind of chugs along all through life, you know, doesn't do much. Here's one that's really, really prominent, you know, at E11.5, and then kind of dies down after birth. We have some that are the opposite, that, hey, look at, I'm not really around much in utero, but once I'm born, and maybe once that person starts talking, look at, it's responding, and the fibroblasts just explode. That gene expression, that's the one that is really doing all the work for an adult. We also can look and see what those fibroblasts are expressing. You know, and in this case, we have different piezos, which are kind of hot right now. Those are like chemosensory, they sense mechanotransduction, collagen. And we can look at like all the different expressions between, here's for this particular HOX-A5 gene, which is really important for development. It's really prominent early on in the fibroblasts, and then later on, not at all. So, and we can look at all the different kinds. So we can do that again for every cell. So we found 23 types of cells, but within those clusters, we found, within each cell type, we found many other kinds. And we have to go through that still to see if there's any distinct cell. But, you know, for fibroblasts, for example, most people are aware of, there's the belief that, you know, the fibroblasts in the anterior and posterior ends of the vocal fold may be different and may be producing different things. There's a whole bunch of literature on that. Well, that actually may be true. You know, whether or not that's, I don't believe that's a stem cell per se, but I believe that functions differently, probably because of the mechanical forces that has. Fibroblasts that are in mid-membranous undergo very different mechanical forces than the ones at the end. We all, you know, and we're going to be able to figure that out. The next step, and I can do this with each of the cells, and we found many different kinds. But the next step is to do, and which we've started in the lab, is to do spatial. And spatial allows us to take each of those individual cells. So this is a nice schematic from a blastoma in my lab, which here's what you just saw, which is a single cell. We took it, and we can say these are the cells en masse. But the next is we can take this and we can use it using gene expression, say, this is actually what this cell is actually expressing and where it is. So that's the next step. So this is kind of, single cell is every cell. Spatial tells us where those cells are. So now I'll be able to say and look and say, if there's really 6 different types of fibroblasts, where are they, and are they located in different parts? And that's, as you can imagine, if you're trying to regenerate tissue, that's super important to know. So we've just started this in the lab, and it's even more expensive. It's kind of crazy, this stuff. So, where's my program? So yeah, here's a great. So here's, we're going from a single cell gene expression to a spatially resolved. This is from Visium, which is the company, and you actually take a section of tissue, it's like an H&E, and then you run it through a lot of stuff I don't understand, and then you actually can get where these cells are actually in the tissue. And I'm expecting to get very different expressions as we think about the idea that the adult vocal fold is potentially, I mean, there's different expression, there's different layering. I suspect, for example, the fibroblasts are going to be very different across that entire dimension of the vocal fold. So, I have 48 seconds. So, okay, so what are the take-homes from all of this? The take-homes that tissue engineering of the laminar propria and the vocal fold is super complex. And, but using the most, hot off-the-press technology, single-cell spatial, we can try to leverage that to comprehensively profile the tissue. We have to put all of that data in free public entities so that anyone in the world can gather that and use that, which helps with the practical challenges of this area. It helps understand the microenvironments to a greater degree. It allows us to study those and make those cells of choice more specifically and easily. Just an overall, much better general grasp of the biology, and may help us answer other questions that have, the holy grail is, why do the vocal folds layer? Why do we get this dimensional change in what's being produced? This data will be able to help us with that. And we'll be able to integrate these atlases. We can do this on intertissue, for example. We're actually, one of my other PhD students is doing this, looking at a mouse that's been injured and how does the cells change over time and what are those? And looking at different healing outcomes, etc. And as with everything, the price always goes down. So right now it's super expensive, but 2 years from now it's going to be actually probably a lot cheaper. So again, this really, all of the work was Ted Lunga's. He is my amazing, he just finished. Ted is just got his PhD, and he is matched and is going to do an otolaryngology residency at the University of Iowa, and I couldn't be more proud of him. All the work I showed you was his. The lab that I use, or our statistician, Christina Kandursky, has been a colleague of mine my entire time at Madison, and her doc student, Elliot, and postdoc Titrison helped with all the data analysis. And everyone else in my lab who are valuable and create a very positive, pleasant environment for science. So thank you. Again, I'm tremendously honored and I greatly appreciate it. Thank you.